Hello everyone, myself Nisha Shah, student of MSc Microbiology second semester, is here to represent my topic post-transcriptional modifications. These are my content. Introduction. The process of transcription involves the production of RNA molecules from the DNA molecules, and what this process actually involves is the passing down of the genetic information from the DNA to the RNA molecules, where these RNA molecules are then directly involved in the process of protein synthesis that takes place in the cytoplasm of our cell. One of the most important type of the RNA molecule is known as the messenger RNA or the mRNA, which actually serves as a template for the protein synthesis that takes place in the ribosome in the cytoplasm of the cell. Before our mRNA leaves the nucleus and enters the cytoplasm, wherein it attaches to the ribosome and goes for the protein synthesis, our mRNA has to undergo several important types of modifications, which include several post-transcriptional modification processes. Thus, the nascent RNA, which is also known as the primary transcript or hnRNA, needs to be modified to become a functional tRNA, rRNA, and an mRNA. These modifications are critical only to the eukaryotic system and not to the prokaryotes, because in prokaryotes the process of transcription and translation occurs simultaneously. The hnRNA, which is also known as the heteronuclear RNAs, are larger than the mature mRNA by many folds, and through post-transcriptional modifications they can be converted into a mature mRNA. This figure shows a double-stranded DNA which is undergoing the process of transcription, resulting to produce an mRNA strand. So as soon as the 5' end of the mRNA is synthesized, it is capped by the capping enzyme, resulting to the first post-transcriptional modification. The second post-transcriptional modification occurs at the 3' end, where multiple adenine residues are attached. Finally, all the introns are removed and all the exons are ligated, resulting to produce a mature mRNA. Therefore, we can say that the post-transcriptional modification includes the three main steps. The first is your RNA capping, second is your polyadenylation, and the third is your splicing. Now let's study each of these steps one by one. So the first is your capping. Capping is defined as the addition of 7-methylguanosine at the 5' end. It is mediated by the capping enzyme, which has a bifunctional activity, that is a 5' triphosphatase activity and a guanyl transferase activity. Now let's look what's the need for capping. So capping probably protects our mRNA from degradation. It also serves as a recognition site for the ribosomes so that the translation may occur, and finally it helps in the transportation of your hnRNA from the nucleus to the cytoplasm. Now let's study about the mechanism of the capping. So in the nucleus of the cell, the 5' end of the pre-mRNA is altered by the attachment of a guanosine nucleotide through a 5' triphosphate linkage. The guanosine nucleotide is then quickly methylated at the 7th position to form a 7-methylguanosine. So this is your mRNA with your 5' end, and here the first enzyme comes into play, which is a 5' triphosphatase, which removes the phosphate group from the 3' end, and then another enzyme, which is known as your guanyl transferase, transfer the guanyl group at that end. Now this guanyl nucleotide will undergo a quick methylation by the enzyme known as S-adenosine methionine, which will result to produce a 7-methylguanosine. This figure shows a special type of linkage that is obtained during the capping procedure, and that linkage is known as a 5' triphosphate linkage. And why is it said to be a triphosphate linkage? As you can see from the structure, 3 phosphate groups are involved in this procedure, and hence it is called to be as a 5' triphosphate linkage. Now let's study about the second post-transcriptional modification, that is tailing or polyadenylation. It is the addition of the poly-A residues at the 3' end, which occurs in the nuclei by the enzymes like poly-A polymerase and CPSF, which stands for cleavage and polyadenylation specificity factors, which together attaches poly-A tail generated from ATP. So this figure shows a eukaryotic mRNA with your 5' end cap and your 3' end polyadenylated. So after the post-transcriptional modification of polyadenylation, what you get exactly is an mRNA having the exons and introns with a 5' end cap and a 3' end polyadenylated. Now let's study how exactly the polyadenylation occurs at this site. So this is your double-stranded DNA, which undergoes the process of transcription, resulting to produce an mRNA strand. So as soon as the 5' end of the mRNA is synthesized, it is capped by the capping enzyme. And for the 3' end to get polyadenylated, there are certain sequences which are recognized by certain factors. And after their recognition, there occurs an endonucleolytic cleavage in between these sequences, due to which we get a pre-mRNA molecule which is capable of undergoing the process of polyadenylation by the help of an enzyme known as poly-A polymerase. So this poly-A polymerase attaches multiple adenine residues at the 3' end. Therefore, we can say that in this type of post-transcriptional modification, the 3' end of the pre-mRNA is removed and a series of adenosine nucleotides are being added at this end. Approximately 200 to 250 adenosine nucleotides are added to the tail. Now, what's the need for polyadenylation? Just as the capping provides stability and resistance against degradation, the polyadenylation or the poly-A tail also provides the mRNA with the stability and keeps the tail away from the degradation. Now let's study exactly the mechanism of polyadenylation. It includes firstly the recognition of the poly-A signal, then a cleavage occurs at the poly-A site, and then a slow polyadenylation is followed by a rapid polyadenylation. This figure shows a complete polyadenylation mechanism. So this is your pre-mRNA molecule with your 5' end which is capped, and there are two poly-A signals. One is your AU-rich signal and the other is your GU-rich signals. So these two signals are firstly recognized by certain factors like CPSF, which stands for cleavage polyadenylation specificity factor, CSTF, which stands for cleavage stimulatory factor, CF1, which stands for cleavage factor 1, and CF2, which stands for cleavage factor 2. So as you can see in this figure, when these factors recognize their respective poly-A signals, it tends to bend your pre-mRNA strand. Then another enzyme called poly-A polymerase binds at the poly-A site, causing the cleavage and the removal of the GU-rich sequence along with their factors like CSTF, CF1 and CF2. This PAP also causes the polyadenylation by using the ATP molecule. However, this polyadenylation occurs slowly. So as to increase this polyadenylation rate, another enzyme called PAB2, that is the poly-A binding protein 2, comes into play. This causes a rapid polyadenylation and also ensures that multiple adenine residues to be added at the 3' end. And whenever it gets the ensurety that multiple adenine residues have been added at the 3' end, it causes the process of polyadenylation to terminate. Therefore, we can say this PAB2 has mainly the two main functions. The first, it causes a rapid polyadenylation, and the second, it causes a polyadenylation termination. Now let's study about the third post-transcription modification, which is splicing or the splicing of exons. Not all the regions of the pre-mRNA molecule code for proteins. Those regions that do code for your protein are known as exons, while those that do not code for your protein are known as introns. Therefore, before the pre-mRNA exit the nucleus, a special protein together known as spliceosome will cut out all the introns and splice together the exons. Therefore, we can say that splicing is defined as the highly precise removal of intron sequences, which is performed by snRNPs and spliceosome. They recognize specific exon-intron boundaries and splice exons together by the transesterification reaction. So this is your 5' exon and this is your 3' exon. Between the two exons, there is an intron sequence which has an A, that is adenosine branch site, which is important for the transesterification reaction so that this intron sequence could be removed or spliced. Now the junction between the exon and intron towards the 5' end is known as the 5' splice site, and the junction between the exon and intron towards the 3' end is known as 3' splice site. Now we will study about the exact mechanism of splicing in the next video.